After the lasers have warmed up and the inline sheath filter has been cleared of bubbles, we will come back to the computer and open the Diva software. For the Diva software, you will have a unique username and password created for you when you pass the practical test. When we log into the Diva software, the first thing that we should do is look on the bottom right-hand corner of the software. There you should see a small yellow dot and text saying Connecting, or a small green dot with text saying Connected. This means that the cytometer is either connecting to the computer or has already connected to the computer. If the dot is black and the text says Disconnected, this usually means that you did not wait long enough between turning on the instrument and turning on the computer. If this happens, you will have to turn the computer and instrument off, wait for 30 seconds, and restart from the beginning. Once the cytometer has connected, we will usually see a pop-up window that will say CS&T mismatch. This means that between the last time someone logged in with this user account and us logging in today, the CS&T beads have been run and new optimized instrument settings have been obtained. If this message pops up, always click the Use CS&T Settings option. Do not click on the option to keep previous settings, as this could adversely affect your experiment. If you are the first person starting the instrument, you will need to run the CS&T beads before beginning your experiment. CS&T stands for Cytometer Setup and Tracking. The CS&T beads will be used by the CS&T software to set the proper laser time delay and to set what it considers to be the optimum voltage for each detector. These beads will also act as a quality control mechanism for the instrument and should be run every time the instrument is turned on. Once they have been run, the CS&T beads should not need to be rerun for the rest of the day. The first step to running the CS&T beads is to dilute the stock concentration of beads to a working concentration by adding 1 drop of beads to between 400 and 500 microliters of saline in a FACS tube. When making the dilution, we need to be sure to label the FACS tube with CS&T, the lot number of the CS&T beads, and the date on which the dilution was made. If you have 200 microliters or more of leftover beads at the end of the run, they can be stored for use on another day by placing them back in the refrigerator. If they are properly stored, the beads can be reused for up to 3 days after they were diluted. But they should not be used after the 3rd day. If there is a FACS tube already containing diluted CS&T beads in the box, check the date on the tube to see if you can use them. If it has been more than 3 days since these beads were diluted, you should throw them away and make new beads. To run the CS&T beads, first we will go to the cytometer menu and click on the CS&T button. This will open the CS&T software, which will connect to the cytometer. While the CS&T module is connecting to the cytometer, we can check that we have the correct lot ID and the correct cytometer configuration. Once we have checked that we are using the correct lot of CS&T beads and that the cytometer configuration is correct, we can load the tube of beads onto the sample port and click the Run button in the CS&T software. Note that you must run the CS&T beads using a low flow rate. A warning message will appear asking us to make sure that we are using the correct bead lot and that the sheath tank is full and the waste tank is empty. Since we already checked this previously, we can press OK. Once we press OK, the CS&T software will then go through an automated process to set up the CS&T. We can see that for each laser, there are only 2 detectors shown, but the CS&T will set the optimum voltage for all of the detectors. We can see a large bead and a small bead population appear on the blue laser forward scatter and side scatter detectors. For the rest of the detectors, the events will appear in the bottom left-hand corner as background noise at the beginning. As the CS&T finds the proper settings, populations of beads may appear and disappear. Eventually, we will see both the small bead and large bead populations appear in the blue lasers forward scatter and side scatter detectors, and 3 populations of beads for each of the other lasers detectors. We should see distinct populations of bright, middle, and dim beads for the red, violet, UV, blue, and yellow-green lasers. The CS&T software will set what it considers to be the optimum voltage for each detector based on these 3 populations, where the bright beads are on scale, but the dim beads are still able to be resolved above the background noise of the system. The entire process can take approximately 7 to 10 minutes to complete. Once the cytometer setup is finished, a message will pop up telling us to remove the tube. After we have removed the CS&T bead tube and replaced it with deionized water, we can click OK. Then another message will come up telling us that the setup has completed. We should always view the CS&T report to double check that everything is working properly, even if there are no error warnings from the CS&T software. The CS&T report will look something like this. There's a lot of information in these reports, but we will only be focusing on a portion of that information. First, we will look at the bright bead percent robust CV column. The CV is a measurement of the variance of the data, and the smaller the CV value, the smaller the variance within the data. We want to check that all of these values are at less than 6%. As we go down the list, we may see that some values are above 6%. This is not unusual for the UVA, UVB, and violet A detectors. If the UVA, UVB, or violet A detectors are between 6 and 12%, they are operating within the normal range. If a detector is not operating within the normal range of CV values, this may mean a number of things. Most commonly, this will happen when the instrument has not been properly cleaned prior to shutdown. If you see abnormally high CV values, the first thing that you should do is clean the instrument using the cleaning process that we will discuss later, and then rerun the CS&T beads. This will generally bring the CV values back into a normal operating range. If cleaning the instrument does not improve the CVs, then the detector may need to be replaced. In this situation, we should not use that detector until the instrument has been serviced by a BD engineer and the detector has been replaced. If there is a high CV, but we are not going to use that detector for our experiment, then we can continue with our experiment. However, we must notify the staff so that they can look into the issue. If all of the detectors for a laser have high CV values, and cleaning does not bring these values back into the normal ranges, then it is possible that the laser is not properly aligned with the flow cell. In this situation, you would not want to use any of the detectors associated with that laser, and should notify the staff as soon as possible so that they can schedule maintenance. In this case, all of our CV values are within normal operating limits, so we can move on to the next part of the report. The second column that we want to look at on the report is the delta PMTV column. This will show us the PMT voltage change between the first time that the staff ran this lot of CS&T beads and today. Ideally, there will be no change, and this value will read 0. However, slight changes in the PMT voltage are normal. If the delta PMTV values are between negative and positive 50, then we can proceed. If the values are outside of that range, the software should give us a warning. Similarly to the CV values that we discussed previously, if there is an issue with the delta PMTV values, we should first clean the instrument and rerun the CST beads. If a single detector is still out of range, then we should not use that detector in our experiment until the staff has investigated the issue. If all of the delta PMTV values are out of range for a laser, then that laser may not be properly aligned with the flow cell. In this case, we would not want to use any of those detectors. If the CV and delta PMTV values are all within acceptable limits, then we can continue with our experiment. Once we are finished in the CST software, we can close that software. We will see the Diva software will reconnect, and we should get the CS&T mismatch pop-up window once again. Since we have just found new optimum settings, we will again choose the Use CST Settings option. Now that the instrument has been started up, we have warmed the lasers up for at least 15 minutes, and the CST has been run, we are ready to create a new experiment in the FACS Diva software. First, however, let's briefly go over the windows found in the Diva software. On the far left is the browser window. Here you can access your experiment, create new experiments, create new groups of tubes within an experiment, or add tubes to an existing group. You can also use the browser window to rename experiments, groups, or tubes. Later, we will also use this window for data export. Next to the browser window is the cytometer window. You can see that there are a number of tabs in this window. For this experiment, we will use the Parameters and Threshold tab and ignore the rest. To the right of the cytometer window is the Inspector window. The Inspector window will allow us to do a number of things, including renaming experiments, groups, and tubes, as well as customizing our experiment. Next is the Acquisition dashboard, which we can use to acquire and record our data. We can also set stopping options and determine the number of events that we would like to display on our plots during the acquisition. Lastly is the Worksheet window. This is where we will create plots and gates to visualize and analyze our data. We will go into more detail on each of these windows later as we go through the experiment. The first thing that we will need to do is create a new experiment. There are two ways to create an experiment in the Diva software. The first is to go to the Experiment drop-down menu and click on New Experiment to choose an experimental template or a blank experiment. Alternatively, we can click on the New Experiment button in the browser window of the software. This will create a new blank experiment. When creating a new experiment, it is always a good idea to rename the experiment. We can do this by right-clicking on the name of the experiment in the browser window, or by clicking on the name of the experiment and then using the Experiment tab in the Inspector window to rename the experiment. Once the experiment has been renamed, we will click on Cytometer Settings under the experiment in the browser window. This will open the cytometer settings in the Inspector window. Here we can see a list of detectors for our instrument. If we click on the name of a detector, we will see a drop-down list of all of the names available for that particular detector. Keep in mind that you can only use one fluorophore per detector. To select only the detectors that we are interested in for our experiment, we will click on the first detector to select it. Hold the Shift key and select the last detector on the list. This will highlight all of the fluorescence detectors. We can then click on the Delete button to remove the detectors. Now we can add in detectors one by one by clicking the Add button. Notice that when we click on the detector drop-down menu, now it shows a list of all possible detector names. As we select detectors, they will disappear from this list, and the list will become shorter. For this experiment, we will choose the FACS detector first. Next, we will click Add again to add another detector and choose the PE detector. We will continue adding the detectors that we will use for our experiment, one at a time. In the Inspector window, under the cytometer settings, we will also notice that there is a column for voltage. Currently, this is set to the optimum voltage by the CS&T software. We can also choose whether we want to display our data on a logarithmic or linear scale. For forward and side scatter, these should always be shown on a linear scale, unless we are looking at very small particles such as exosomes or bacteria. For fluorescent markers, we will typically use a logarithmic scale, with some rare exceptions, such as cell cycle analysis, where the G2 population will have precisely 2 times the fluorescence of the G1 population. We can also choose which measurements of the signal pulse that we would like to record. That is, the area, width, and height of the pulse. For BD instruments, it is recommended to use the default area measurement for fluorescence signals. For the light scatter parameters, we will also choose to record the height so that we can do doublet exclusion in our analysis. Once our cytometer settings are properly set up, we will next tell the software that we want to create compensation controls. To do this, we will again go to the Experiment drop-down menu, and this time click on Compensation, and then Create Compensation Controls. This will open a new dialog box, asking us what controls we have. We should have an unstain control, as well as single stain controls for each fluorophore or dye that we will be using in the experiment. For the labels, we will leave these set to generic for now, but we will come back to labeling later. Once we have confirmed our compensation controls, we can click OK. This will create a group called Compensation Controls under our experiment in the browser window. If If you click the little plus sign next to the group, you will see a list of all of our compensation controls. We will also see that the worksheet window has switched from the global worksheet to a normal worksheet. This is important as compensation controls need to be set up using a normal worksheet, whereas analysis will be done using the global worksheet. Next, we will select the unstained control tube from the browser window. Note that even though I clicked on the tube, I cannot acquire or record any data in the acquisition dashboard. This is because I only highlighted the tube when I clicked on it. To actually select the tube, I need to click on the little arrow next to the tube name. When the arrow turns green, that tube has been selected. Now that we have the proper tube selected, we should open the Parameters tab in the cytometer window. This is where we will be able to change our detector voltages as needed for our experiment. Next, I will load the tube onto the sample probe, make sure that the instrument is in run mode, and set to a low flow rate. Then, I will press the Acquire Data button. We should see events begin to appear on our plots in the workspace. Notice that a large number of these events appear on the far right edge of the plot for our forward scatter and side scatter. This means that they are off scale, and the forward scatter voltage will need to be adjusted down. To do that, we will go back to the Parameters tab in the cytometer window and lower the forward scatter voltage by using the slider bar or typing in a number. We will then press Restart in the acquisition dashboard to look and see how our data has changed. In this case, the voltage has decreased too much, so we will go back and increase the voltage until our populations are clear and the events are on scale relative to the far right, top, and bottom of the plot. In this experiment, we are using peripheral blood mononuclear cells and can see a few different populations. There are lymphocytes, monocytes, dead cells, and debris present. We want to put our P1 gate around the live lymphocytes and monocytes while excluding the smaller dead cells and debris. Now we can see the background noise or autofluorescence signal in each of the detectors. We will not worry about the autofluorescence for now, and will mainly focus on the positive signal from our positive controls. Next, we will stop acquisition and load our first positive control, the FACS positive control. We are going to leave the unstained control selected in the software That is, the green arrow will be highlighted next to the unstained control, but we will physically change to the FACS control tube on the sample port. This will allow us to see the FACS signal in all of the detectors and look for potential issues with spillover into other detectors. Once the FACS tube is loaded, we will again begin acquiring data to see what it looks like on our plots. We can see that there is both a FACS positive and negative population in this sample, with the positive population being off scale. Just as with the forward scatter, we will need to adjust the FACS voltage to bring the signal on scale. Note that you will only want to decrease the voltage enough to bring the signal on scale, as decreasing too much will push the positive signal closer to the background noise. That would reduce your signal to noise ratio, which we do not want to do. When running the compensation controls, we want to check two things. First, that the signal is on scale and brightest in the primary detector. In this case, the primary detector is the FACS detector, because that is the fluorophore that this control is stained with. We want to make sure that the signal is brighter in the FACS detector when compared to all other detectors. In this case, the FACS signal is around 10 to the 5th in the FACS detector and less than 10 to the 3rd in all other detectors. Therefore, it is brightest in the FACS detector. Once we have found a voltage where the signal is on scale and brightest in the primary detector, we can stop acquiring the FACS control and do the same with the PE control. We will again leave the software on the unstained control, load the PE tube, and acquire the data. We can see the PE signal is on scale near 10 to the 5th and brighter in the PE detector than in any of the other detectors. Therefore, we can keep this voltage and move on to the other controls. The APC signal comes in at approximately 10 to the 4th in the APC detector and has very little spillover into the FACS and PE detectors. There is, however, spillover into the APC size 7 detector, but since the APC signal is much higher in the APC detector, we do not need to change the voltages and can move on to the APC size 7 positive control. Here we can see that the APC size 7 control also spills over into the APC detector. Because their signal strengths are close, we could increase the APC size 7 detector voltage. As we can see, this will shift the positive signal in that detector to the right. Because we increase the detector voltage, however, we also increase the background signal in that detector. So, we will need to recheck the APC positive control and make sure that it is still brighter in the APC detector when compared to the APC size 7 detector. Now that we have checked all of our controls and have found voltages for each detector that will bring the signal on scale and brightest in the primary detector for each control, we can begin to record our data. Remember, we must keep voltages constant over the entire experiment, so this first step of finding the optimum voltage must be completed before we can begin recording any data. Another thing that we should do before recording is adjust our threshold to remove some of the dead cells and debris from our analysis. To do this, we will go to the Threshold tab in the Cytometer menu, and we can see that there is a threshold on the forward scatter parameter set to 5,000. We can increase this value and load our unstain control to see how this alters our data. We can see that now anything below the 60,000 mark is cut out. We do want to be careful though not to set the threshold too high, as this will cut into our actual cells of interest. Now that our voltages and thresholds are set, we can begin to record our control data. If we want to, we can increase the number of events to record. However, for this experiment, we will leave that at the default 5,000 events for our compensation controls. As the data records, the progress bar will fill up, and once 5,000 events have been recorded, the software will stop recording data. It is important to remember, however, that your sample is still being run through the instrument, even if it's not being recorded. When the sample has recorded the data, a small disk icon will appear for that sample tube. Next, we will right-click on the P1 gate and apply that gate to all of our controls, so that they will all have the same P1 gate. Now, we click on the Next Tube button, or select the FACS control from the browser window. You should always wait at least 4 to 5 seconds before pressing the record button. Next, we will move the P2 gate to include the brightest positive population. When adjusting the P2 gate, make sure to only include positive cells. The P2 gate should never include any cells from the negative population. Once the control has finished recording, we can move on to the next control and do the same thing. We will continue this for all of our single color controls. Note that if there is a middle population between the negative and brightest populations, we will ignore that for the purposes of compensation, and only gate around the brightest population. Once all of our compensation controls have been recorded, and the P2 gates have been properly adjusted, we will need to calculate the compensation. To do this, we will go back to the experiment drop-down menu, click on Compensation, and then click on Calculate Compensation. A new window will open, telling us that the calculation has been completed successfully, and we will choose to link and save the compensation. If we choose the Apply Only option, the data will be compensated in Diva, but will not be compensated in any exported data. Also note that we do not need to rename the compensation. We can leave the default timestamp date. Once the compensation has been applied, we can see a little gold icon appear next to the cytometer settings. This lets us know that the samples will be compensated. Next, we will create a group or specimen using the New Specimen button in the browser window, and name it. Then, we will click on the small plus sign to open that group and see that there is a tube, tube 1, already there. We can select tube 1, go to the Labels tab under the Inspector window, and label our antibody targets. Alternatively, we can go to the Experiment drop-down menu, Experiment Layout, and label the antibody targets from here. This is useful if you have multiple tubes, as we can use the same label for all of the tubes, rather than individually labeling each tube. We can also use the experiment layout to set the number of events to record for each tube as well. For this experiment, we only have 2 tubes, so we will delete the others and rename our sample tubes. Next, we will change from the normal worksheet back to the global worksheet, either by clicking on the Global Worksheets button in the Worksheet window, or by double-clicking on the Global Worksheet 1 in the browser window. We can now begin to add our plots to the global worksheet for our data analysis. We have 3 different plot types: dot plot, contour plot, and histogram. For this, we will choose the dot plot by clicking on it once and then clicking on the worksheet to add the plot. We can also double-click on the dot plot to add multiple plots, 1 per click on the worksheet. Once we have all the plots that we need, we can click the cursor icon to deselect the dot plot creator. The next step will be to choose our parameters for each plot. The first plot will be a forward scatter area versus side scatter area plot, used to find our cells based on size and complexity. For the second plot, we will choose side scatter height versus side scatter area, or forward scatter height versus forward scatter area for doublet exclusion. Note that we could use 2 separate plots for doublet exclusion, but for this experiment, I'm only going to do 1. For the following plots, the parameters will depend on the experiment setup. In this experiment, we are looking for CD4 and CD8 T cells, so we will first plot CD3 versus CD14 to find our T cells. Then, on the next plot, we will look at CD4 versus CD8. Again, for your experiment, the parameters will be different, and this step will depend on your eventual gating strategy. We can use the unstained cells from our compensation controls to set up the first few gates. There are a number of gate options, including polygon, rectangle, quadrant, and histogram. For this first cell gate, we will use a polygon gate. For the polygon gate, every click creates a vertice in the polygon. I can double-click to close the polygon. Now that our cell gate has been drawn, we can click on the next plot and make sure that it only shows the events in that gate by either clicking on it, going to the inspector window, and clicking on the population P1 check, or right-clicking inside of the plot and choosing to show population P1. Next, we want to draw our doublet gate. To do that, we will first enlarge our plot, then use a polygon gate to cut out our doublets. Remember, doublet cells have a greater area to height ratio, so we want to exclude cells above that 45-degree line. Once our gate is set, we can make the plot smaller again by resizing it. Or by holding the Shift key and selecting another plot, then clicking on the Make Same Size button. This will resize the plot to the original size. Next, we will go to the third plot and make sure that it is only showing single cells by showing population P2. For this plot, we can use rectangle gates to create a T-cell and monocyte gate based on our CD3 and CD14 expression. For our final plot, we again want to only show CD3 positive T cells. Note that there are no cells showing up in the final plot right now, as there are no cells in the CD3 positive gate in our unstained control. Now we can select the sample that we want to run, but before recording, we should decide if we want to set a stopping gate. By default, the stopping gate is set to all events. So in this case, we would collect 5,000 total events. But we could set it to collect 5,000 events in our P1 cell gate, our P2 single cell gate, or any of our other gates. It is important that the smallest population of interest in your sample has at least 2,000 events for statistical analysis. So, if we were only interested in CD3 positive cells, we could set our stopping gate to P3 and our events to record to at least 2,000. Also note that our storage gate should always be set to all events. The storage gate is the data which will be recorded, and we always want to record all of the data. For this experiment, we will collect 20,000 single cells. Once our stopping gate and events to record are set, we can load our sample tube and begin recording the data. Notice that the threshold count will be different from the stopping gate count. This is because threshold count represents all events. Also note that the events per second should not exceed 10,000. Once our unstained sample has been recorded, we can move to the next tube, our stained sample. Notice that the events to record needs to be changed again because I did not set this up in the experimental layout at the beginning. When we begin acquiring this data, we can see that the P3 and P4 gates are not set properly. This is because the unstained cells are not a good gating control. We will need to readjust the P3 and P4 gates to only gate around the CD3 T cells or the CD14 positive monocytes. When we look at our fourth plot, we can see that there are events jammed up against the X and Y axis. To bring this data back on scale, we will highlight this plot, go to the inspector window, and choose the bi-exponential display for these axes. We can now see all of the data. We can also see that there is some unused white space on the plot. To fix this, we can go to the View menu and choose the bi-exponential editor. With the bi-exponential editor, we can choose to adjust the scaling manually and change the numbers for the scaling for each fluorophore to remove some of that white space. Once the plots look how we want them to, we can choose to apply the values to the experiment so that all the samples will have the same scaling. Now that we can see our data, we can create a quadrant plot for the CD4 and CD8 T cells. Notice that we have double positive cells in Q2. This is likely because I did not include a viability stain in this experiment, and I am picking up some dead cells, which are non-specifically binding to the CD4 and CD8 antibodies. This is why it's always important to make sure to include a viability stain. If I want to add more data to my sample, I can choose the sample tube from the browser window and then press record again. This will give me the option to overwrite the previous data or append the new data to the already recorded sample. In this case, we will append. Once we have run our last sample, we can begin the process of cleaning the instrument. The cleaning will take approximately 15 to 18 minutes. So we can begin cleaning while analyzing and exporting our data. To clean the instrument, we will use 3 cleaning solutions: FACS Clean, or 10% bleach, followed by FACS Rinse, followed by deionized water. To start, we will load a tube that is half filled with FACS Clean onto the probe and leave the arm to the side for 1 minute. After that 1 minute, we will move the arm underneath the probe and let the instrument run on a high flow rate for 5 minutes. We will then repeat this process with the FACS rinse and the deionized water. After the deionized water has finished running for 5 minutes, we will make sure that there is less than 1 milliliter of water in the tube and put the instrument on standby mode at a low flow rate. While the instrument is cleaning, we can adjust our worksheet and show the data how we want. Then we can export the data. If we right-click on any of the plots, we can then click on Show Population Hierarchy. From here, we can rename the different cell populations and change their colors in the plots. We can also adjust where the population names fall on the plots. If we want to, we can also click on a plot and change the background color in the inspector window. This can make certain colors easier to see. Likewise, in the inspector, we can change the number of events to display on a plot, which can sometimes make gating easier. We can also customize the plot title and adjust fonts and font sizes if necessary using the inspector. We can use Diva to do basic statistical analysis by right-clicking on our plot and creating a statistics view. This creates a statistics box, which we can customize. Generally, I like to remove all of the header information, then choose the populations of interest, and finally, the statistics that I want to look at. In this case, we're going to look at the MFI of CD4 on CD4 and CD8 positive T cells. We can also increase the number of worksheet pages if we need more space for additional plots using the inspector window. Once the worksheet is set up how we like, we can then begin the process of exporting the data. First, we will right-click on the name of the experiment in the browser and go to the export window. From here, we can choose what we want to export. The experiment file is a Diva file, which will have all of the data and gating, but can only be opened in the FACS Diva software. The FCS files are raw data files, which can be opened for analysis in any flow cytometry analysis software, such as FlowJo or FCS Express. But the FCS files will not contain any of the gating information. Lastly, we can export a template of the experiment if we will be repeating this experiment at a later time point or with different samples. Today we will start by exporting a template. When we click on this option, the template wizard opens. All we have to do is give the template a name and choose the type. The type will correspond to your lab and principal investigator. I can then click Next to add more information, or simply click Finish. Now that I've created the template, I can find that template to create a new experiment by going to the Experiment drop-down menu and clicking on the New Experiment option. Next, we will export the experiment file. We want to make sure that this exports as a zip file to conserve size. We will then browse to where we want to save the file. I will save the file on the local D drive. I can also export the FCS files. For these files, we want to make sure to export the most recent version, and then press OK. Lastly, we can export our worksheet as a PDF. If I only want to export one sample, I can use the Make PDF button at the top of the worksheet window. Otherwise, I can select my first sample in the browser window, then right-click on the name of the group and choose Batch Analysis. If I have more than one group in the experiment, I should right-click on the name of the experiment and choose Batch Analysis from the experiment in order to export all of the samples to the PDF. Next, I will choose Save as PDF. I can also choose to add a report page, which is basically a cover page. I could choose to export the statistics from my statistics box into an Excel readable file as well. I will then want to freeze the by exponential scale so that all of my scales will be the same. Since I only have one worksheet, the preferred global worksheet can be checked or not. It won't really matter. Using more than one worksheet is a little too advanced to go over in this course. Next, I will use the Browse option to set where I want the PDF file to save, and then press Start. Once the samples have been processed, a message will display telling us that it is finished. If we look at the report, we can see the cover page, and then our two samples, each on an individual page. Once the data has been exported, we can right-click on the name of the experiment and delete it from the browser. This is an important step, as too many experiments in the software will cause the software to slow down for all users. Once the browser has been cleared of experiments, I can log out of Diva by going to the File menu and choosing Log Out. Do not quit or close the software before logging out, as this may not log you out of the software. Once you have logged out of and quit the Diva software, you can log off of the computer. If you are the last user of the day, you will shut down the computer instead of logging out. Once logged out of the computer, if the cleaning procedure has not been finished, we will continue the cleaning process. If you are not the last user of the instrument for the day, you will leave the instrument on standby mode at a low flow rate with less than 1 milliliter of deionized water in the tube. Please also remember to clean up the area after you are finished, and do not leave behind any tubes, pipette tips, or other lab waste.